Now we will start with immunomagnetic sorting of plasma cells. The equipments and consumables which are required for this procedures are 15 ml conical bottom falcon tube, 12 ml round bottom tube, micropipettes 20 to 200 microliter and 100 microliter to 1 ml. Sterile tips, centrifuge, swing bucket with minimum 1000 revolutions per minute. Stem cell magnets. Stem cell PBS buffer. Stem cell RBC lysis buffer. EasySep CD138 cocktail, EasySep whole blood magnetic particles. And lastly, freshly made Carnoy's fixative. Before starting the procedure, all the consumables should be brought to room temperature. The first step is to take minimally diluted bone marrow aspirate sample 2 to 3 ml, preferably from the first pull, in a green top vial containing sodium heparin as anticoagulant. Assess the sample for any clot. Take extra care that sample is minimally diluted. Also note that the sample should be brought to room temperature before the start of processing. Transfer 1 to 2 ml of heparinized bone marrow sample to a 15 ml conical bottom falcon tube. Now add stem cell PBS or potassium-free PBS buffer. The stem cell PBS buffer is added. The heparin vial can be washed thoroughly with the same buffer, and the entire contents are now transferred to 15 ml falcon tube. Now the final volume is 10 ml. After this, centrifuge at 200 G for 10 minutes. After the centrifugation, discard the supernatant. The supernatant is discarded. Leaving buffing coat of approximately 0.5 ml above the pellet. Please take care not to disturb the pellet. Now add stem cell PBS buffer. The stem cell PBS buffer is added to make the volume of 2 ml if required. To this, add equal volume of 2 ml stem cell RBC lysis buffer to make a final volume of 4 ml. Now, the entire contents are transferred to a round bottom tube. We can use around 1 ml of PBS buffer to thoroughly rinse the falcon tube and transfer the contents to the round bottom tube. After transferring the contents, mix well and gently with the help of a pipette. The next step is to add CD138 cocktail. Gently mix CD138 cocktail with a pipette . Now add 50 microliter to the sample contents. 25 microliter can be added for each 1 ml starting bone marrow sample volume. CD138 cocktail is now added. Mix the contents gently with the help of a pipette. Now gently cap the tube and incubate at room temperature for 5 minutes without disturbing the tube. After 5 minutes incubation, next step is to add stem cell magnetic beads. Mix the stem cell magnetic beads thoroughly by gentle pipetting up and down for 25 to 30 times till the beads are uniformly dispersed. Now add 50 microliter of beads to the sample mix and mix the contents gently. Incubate the tube at room temperature for 5 minutes without disturbing the tube and gently capping it. After the 5 minutes incubation, gently mix the contents. Now place the tube in the magnet for 5 minutes without capping it. Please take care that the magnet should be kept away from any kind of vibrations. After a period of 5 minutes, discard the supernatant. The supernatant is discarded without removing the tube from the magnet by tilting the magnet with the tube upside down. Be cautious to dab the extra drops at the rim of the tube with the help of a clean tissue paper. Now add 5 ml stem cell PBS buffer directly in the center of the tube. Take care not to disturb the walls of the tube. Leave the tube for another 3 minutes without disturbing the magnet. Do not cap the tube. After 3 minutes, discard the supernatant. The supernatant is discarded. Gently remove the tube out of the magnet. After this, again add 5 ml stem cell PBS buffer. The stem cell PBS buffer is added to thoroughly rinse the sides of the tube. The contents are then transferred to a 15 ml conical bottom falcon tube. Now add 5 ml freshly made carnoise fixative to the round bottom tube. Again rinse the sides of the tube thoroughly. Now transfer the contents to the 15 ml falcon tube. The final contents of the tube are 10 ml. This is followed by centrifugation at 200 G for 10 minutes. After the centrifugation, Discard the supernatant. Look for the cell pellet. The cell pellet is dislodged and 10 ml of freshly made carnoise fixative is added. Now again centrifuge at 200 G for 10 minutes. After the centrifugation, take out the tube and observe for clear pellet. The last step is chilled fixation. Keep for overnight fixation at 4 degrees Celsius after labeling the tube with patient details.